Thank you for standing by and welcome to the Arovella Therapeutics Investor Webinar and Q&A today. All participants are in a listen-only mode. There'll be an initial slide presentation lasting for approximately 30 minutes, followed by Q&A based on questions we've received by email and any that come through live. So if you do have a question you want to submit live, please feel free to type it in using the Q&A function on the Zoom app. But appearing on behalf of Arovella today, we have the CEO and Managing Director, Dr. Michael Baker. Michael, I'll hand it over to you.
Great. Thanks, Matt. Appreciate the intro and thanks for setting up the webinar. And thanks everybody for taking the time to dial in and listening to the presentation today. I'm looking very much forward to taking you through Arovella as we've come over the last 12 months, but of course where we're headed, and looking forward to updating those that already are familiar with the company, but certainly welcoming those that are less familiar and joining for the first time. So I will be making forward-looking statements, so please do take note of our disclaimer. Now, I'll just jump straight into the highlights of the company. So what we're working on is what we call an invariant natural killer T cell, or iNKT cell platform for short. And we're using that to tackle a range of different cancer types, including both blood cancers and solid tumor cancers. And our leading product is termed ALA-101, and it's a potential treatment for what we call CD19 expressing blood cancers. And we're expecting that to commence or progress into phase 1 clinical trials in 2024. So we do see that the cell therapy sector itself has really made waves in terms of cancer treatment. And we think that our iNKT cell platform actually solves a number of key challenges that have hampered the cell therapy sector to date. Now, in addition to our platforms and our technologies, we've worked equally hard to build a very strong leadership group, both at the board level and management level. So we do have a lot of experience in drug development and particularly around the development of cell therapies. Now, over the last couple of years, we have worked hard to acquire new technologies, but we'll certainly continue to keep that as part of our DNA, looking to find technologies that we think either strengthen our cell therapy platform, but also align with our core focus areas. Now, we are one of a handful of companies globally and the only company in Australia working on iNKT cell therapy. So we do think that we are actually uniquely placed, and that positions us well for growth. Now, in terms of our financials, we're currently valued at about $42 million. And as of March 31st, we had about $7.1 million cash in the bank. So we're delighted that we recently closed an oversubscribed placement to raise $4.1 million. And I will point out, we also have a share purchase plan currently underway to raise an additional $1 million. So we are looking well in terms of our cash position. And we're delighted that over the course of the first 6 months that we have had quite a strong appreciation of our share price, which we think is off the back of a number of key results following from our data presentations, but also some strategic and well-recruited individuals into the company. Now, in terms of our shareholders, we're delighted that Merchant is our largest shareholder, and they're very supportive of the company and what we're doing and the therapeutic strategy. And we've also got a number of other high net worths and also smaller institutions that have been quite supportive of the company and the activities that we're undertaking. Now, in terms of the last 12 months, we have had a very busy period, and I think there's quite a few things to highlight, but I'll focus on a few. I think, as I mentioned a moment ago, building a really strong team has been important. So delighted to have Gary Phillips join as a NED. Excellent to have Dr. Nicole van der Veen appointed as our COO, who's making a great contribution to the company. And Dr. Tom Duthy joined recently as our new non-executive chair. Now, in terms of our platform, we were delighted to have our European patent granted, which covers the iNKT cell therapy platform itself. We've also entered into an option agreement with the University of North Carolina, which we think provides the ability to enhance our iNKT cell therapy platform. And in terms of validation, delighted that we started a strategic collaboration with Imugene. Earlier this year, we received positive in vitro data for that collaboration, and we're looking forward to getting more data in animal models in the second half of this year. Now, in terms of our lead product, ALA-101, we made significant progress on generating the components required to manufacture the drug. And we presented data that was using that material at a very well-regarded oncology conference, the American Association for Cancer Research, in April. where we presented both data confirming in vitro, in vivo activity of the therapeutic, and we believe that was a big trigger for part of the share price accretion that we saw earlier in the year. So in terms of the landscape, I think it's been hard to avoid the last 24 months, and a lot of people would recognise it's been a pretty difficult time for the sector. But what we've seen over that period is still some pretty sizable transactions. And as I mentioned a moment ago, we're working on ALA-101. We're currently at the preclinical stages. And what's important to note here is that a number of those deals that we have seen are in the preclinical stages for cell therapy companies. And in addition, there's quite a number at early clinical stages, such as phase 1, that you can see here as well. So just reiterating, Arovella is currently at the preclinical stages, but looking to get into clinic in 2024. And when you look at the upfront payments for some of these licenses or acquisitions, you can see that ranging from 70 to 245 million, they are quite sizable without even taking into account milestone payments or total deal value, particularly given our current market cap is only about 42 million. So we do see that there's a lot of scope for additional value creation. So just stepping back to what we do, I'll explain what cell therapy is by using what's more well known to date, which is CAR-T cells. So this is what I almost refer to as generation 1 of the therapy. And the way it works is blood is actually taken from a patient's arm. And so this is the patient that will actually have the particular form of cancer to be treated. We collect the T cells or the immune cells. They get genetically reprogrammed in a laboratory. So they actually produce what are called chimeric antigen receptors, or CARs. I'd like people to think about these as almost like heat-seeking missiles that then decorate the outside of the immune cell, and they'll actually drag the immune cell to find and kill the cancer cells once it's in the body. So they get grown up into the millions and then infused back into the patient that has the particular form of cancer. So I said, once they get in, they recognize the particular marker on the surface of the cancer cell and they trigger its destruction. Now, it has, I mentioned a moment ago, it has revolutionized blood cancer treatment. And that's largely because we're now at a point where we're using the words cure and cancer in the same sentence. And there was a series of articles that came out over February 2022 and May 22 in the adult and pediatric settings, where patients that were taking these types of therapies 10 years ago are still cancer-free. So that's why we're so excited by it. It's got transformative potential in that sense. Now, in terms of the market size, not surprisingly, with that level of activity, it's expected to reach north of 60 billion by 2030. And for the two earliest approved products, Yescarta and Kymriah, both, one did over 1.1 billion, the other over $500 million in revenue in 2022. And another one that was more recently approved, Abecma, which was approved in 21, did just under 400 million. So it's, again, a very promising sector that is set to get a lot bigger. Now, we see that there are some particular roadblocks with the cell therapy sector as it stands. And that's largely off the back of using the T cells that all 6 products have now been approved. So for those therapies, the T cells must originate from the patient to be treated. And in that sense, it means that every single dose is made bespoke for that individual, which the pharmaceutical industry wasn't really ever set up to operate under those sorts of conditions. So this is net result have high manufacturing costs, increased supply chain, logistics and issues. It also means the starting material must come from the person that has the disease, which those diseases are known to compromise the immune cells, so it can reduce the efficacy. And also not all cancer treatment centers are actually able to collect and manufacture. So it means that some patients that may actually be eligible to take these therapies are not in the position to be able to get access to them. Now, in addition, it takes about 4 to 6 weeks for these therapies to be generated. So patients with a particularly form of the disease, they can actually succumb to the disease while they're waiting for their treatment. But of course, and this does happen, unfortunately, while it's being manufactured for every single person, it opens up the opportunity for a manufacturing run failure, which again increases time and cost. So we do see that our allogeneic or off-the-shelf iNKT cell platform has the potential to address some of the manufacturing logistics challenges, but also to have the potential for a product with improved efficacy as well. And I'll talk more about why we think that's the case, starting with the current therapies that are the more well-known, which are both T cells and NK cells. So T cells, they are known to have some pretty nasty side effects, such as cytokine release syndrome, as well as neurotoxicity. And in order for them to be given from a healthy person to another patient, let's say, so we could use them off the shelf, they do require complex gene editing. And for the NK cells, while they can be given from a healthy person to a patient, we have seen that there's potential for limited persistence, and that may also limit the durability of the response from those cell types. So that's in contrast to iNKT cells, which are unique. They actually bridge the adaptive immunity and the innate immunity, and they take properties of both T cells and NK cells. Now, really importantly for us, they can actually be given from a healthy donor to many patients because they don't cause what we call graft versus host disease. So that's been clearly demonstrated now in clinic for other iNKT cell types. So we're quite comfortable that we can accelerate with an off-the-shelf product. Now, through this little red bump here called the invariant T cell receptor, they actually naturally target and kill certain cancer types that produce something on their surface called CD1D. And so when we arm them with the chimeric antigen receptor, or CAR, they become dual targeting. So there's 2 shots on goal. Now, they also take the killing mechanisms from both T cells and NK cells. And in contrast to the other two, they can actually modify what we call the tumor microenvironment that actually causes a pro-tumor or pro-cancer state. So iNKT cells have that effect, not T cells or NK cells. They also infiltrate tumors, and they can produce other molecules or signals that tell T cells and NK cells to start destroying the cancer cells as well. Now, in terms of the commercial landscape, this was also an important factor for why we started working on iNKT cells. We look at again, this is a snapshot of logos or companies working on T cells, and same for a snapshot of NK cell companies. And the reality is there's hundreds, if not thousands, hundreds of companies and probably thousands of clinical trials. But when we look at the iNKT cell landscape, what we see is it's still a handful of companies working in this space. So we have very carefully put ourselves into a position where we're operating in one of the most important areas of cancer treatment, and we've carved out a nice niche in that space. Now, in terms of the manufacturing process, this is a schematic of how we would expect it to look. We have a healthy donor with a fully functional immune system. We will collect their blood, and we isolate the iNKT cells, which, as I mentioned, they have that ITCR that can recognize cancer cells. We then engineer them in a lab to produce the chimeric antigen receptor, or CAR. So that's the targeting missile. They get grown up into the billions before ultimately being put into vials and then frozen and stored ready for use, so that when a patient is actually eligible to take the therapy, they can be pulled out of the freezer and given to that patient. So we believe there's a number of advantages. Firstly, you have healthier starting material because the donor has not seen the cancer or any type of therapy like a chemotherapy, which can be quite nasty to immune cells. It's scalable, so we can produce multiple doses per batch. Because we don't have to wait for the therapy to be produced, it has faster access to treatment, and also removes the risk of manufacturing run failure because they are stored in the freezer ready for use. Now, in terms of our pipeline, so we do have a number of products under development or programs under development. Our lead is ALA-101, which we are using to target CD19 producing blood cancers like non-Hodgkin's lymphoma. We're also very excited to be working with Imugene to combine ALA-101 with their onCARlytic virus, which opens up the opportunity to target a range of different solid tumor types. And we've also got our third product, DKK1 targeting CAR, which we have data to suggest will be useful to treat cancer types like multiple myeloma, lung cancers, and pancreatic cancer. So really important point of this slide also is to highlight that we have control over what CAR we put into the iNKT cells. So we currently are screening and looking for other CARs that we think would fit well with our platform, and they may be in the blood cancer or the solid tumor setting as well. So I'd just like to highlight some of the data for why we're so interested in ALA-101 and why I think this has got great potential to be an excellent therapeutic strategy for CD19 expressing blood cancers. And just to give a bit of background, B-cell lymphoma, like non-Hodgkin's lymphoma, is quite prevalent, with an annual incidence in the US and Europe of 65,000 and 95,000 respectively. Now, many patients that are actually diagnosed with the condition that take the first-line therapeutics, which are not cell therapies, 60% or more will actually relapse and require other therapeutic strategies. And when we also look at B-cell leukemias, there's an incidence of about 23,000 per year in the US. So in terms of current treatments, so looking at the cell therapy landscape, there's 4 approved CAR-T therapeutics to date that target CD19. Now, all of them must come from the patient. So there's still no CD19 targeting allogeneic therapy approved as yet. Importantly, the therapeutics strategy is gaining momentum and they are moving up to second-line therapy, which is good. But we still see for the CAR-T approach that a significant number of patients do relapse following CAR-T treatment. In addition, there's also substantial safety risks such as cytokine release syndrome, neurotoxicity, and infection. So we do believe there's a significant unmet need, and we believe that ALA-101 is very nicely placed to fill that need by having an off-the-shelf strategy to treat B-cell lymphomas and leukemias. And we're very excited that we're expecting to have our phase 1 clinical trial commence in 2024. So just to highlight the data that we've generated to date with the CAR-19 targeting iNKT cells, this is just two snapshots of data. One up the top, what we're looking at here is these are animals after 3 days of treatment with a range of different therapies. And anywhere you can see the color, the red, green, blue, it's a way of tracking the tumor cells. So anywhere you see the color, that's actually the tumor cells in the animals. In the first column, it's treated with essentially nothing, with empty immune cells, T cells, empty iNKT cells. And then we've got the CAR-T targeting CD19, and then the CAR-INKT targeting CD19, ALA-101. So I think it's quite clear after 3 days, you can see that there's still quite a large amount of cancer cells persisting in what we call control groups. But for ALA-101, it's demonstrating how quickly and robustly the therapy can work for cancer types that produce CD19 and CD1D. And then what we're looking at on the bottom graph, it's a survival curve. So it's a similar study setup, only now we're looking at for longer time points. So instead of taking this from looking at this at 3 days, we take it all the way out to 90 days. And then we're looking at the vertical axis is looking at how many animals survive over that time period. And so what we can see for what we call as the control groups, they've all died between 40 to 60 days. For the CAR-T cells, so the commercially available type of therapy we see, still very impressive data. It's 60% of the animals alive at 90 days. But it's the CAR-INKT cells at the top here, we see that it's about 90% of those animals are still alive. So again, really encouraging data. It works quickly and it promotes long-term survival, which is exactly what we would like to see in phase 1. And then there's an interesting piece of data that came out of these studies where 4 of the mice that were actually treated with our therapy, CAR-19 iNKT cells, you can see here that the 4 mice all went cancer-free at the earlier points, or looked like the cancer was completely eradicated. And over time, what we saw between, let's say, 20 and 30 days or 20 and 40 days is the cancer cells actually returned, and specifically to the brain in these animals. And so natural response here is that the animals will succumb to the disease. But instead, what actually happened with no further dosing, the animals will actually undergo what was termed a spontaneous secondary remission. So we think that opens up some interesting avenues for us to continue to pursue. One is that we may consider using this for a more neurological type of cancer, because it suggests that the cells, they can remain to kill cancer types like this, they can cross the blood-brain barrier and even have an impact on a more solid tumor-like cancer. So we then, as I said, we presented at Orlando at the American Association for Cancer Research in April this year. And what we were showing is an extension of the data using the product that was now more geared towards going into clinic. And we were able to demonstrate that the iNKT cells can be well expanded, and following their expansion, they can still multiply and kill tumor cells that express CD19. And that once they're stimulated by those cancer cells with CD19, they produce the components that we want them to in order to be able to kill the cancer cells. In addition, they also, we clearly demonstrated they can kill tumor cells isolated from patients that have particular forms of cancer. And we also tested them in a very aggressive B-cell acute lymphoblastic leukemia model that doesn't produce one of the targets, CD1D. So we really made it quite difficult for the iNKT cells, and we still saw a significant extension of lifespan. So just to summarize that data, it's now given us great comfort that our proprietary manufacturing process allows for efficient expansion of the iNKT cells, but retains their functionality. And that's really important when we consider making an off-the-shelf product, so we're able to get multiple doses, so we have the advantages in terms of the ability to scale and, of course, reduce costs. Now, as I mentioned, we'd taken that a step further from the original work, where we started using what we call third-generation lentiviral vector to make the product. And that's really important because it contains the genetic elements or the pieces that actually have a proven safety profile to regulators like the FDA. And as I said, we were able to show that ALA-101 does confer a significant anti-tumor effect and significantly extend lifespan in this particular form of a B-cell acute lymphoblastic leukemia. So we're very, very excited, as we do think that this has a genuine chance of being an effective therapeutic treatment for CD19 producing leukemias and lymphomas. And so we're looking forward to progressing this towards first-in-human clinical trials. And I'd like to quickly just touch base on our ALA-101 and onCARlytics, or CF33-CD19, just to reiterate why we're so excited by this strategy. So it's essentially a combination of the two products to create an off-the-shelf cell therapy that could be used with an oncolytic virus to mark and destroy solid tumors. So I've just shown ALA-101, it is a very potent and rapidly activated to kill CD19 expressing cancers. It is being developed as an off-the-shelf treatment, and onCARlytics, or CF33, I should say, is an oncolytic virus that specifically targets tumor cells. Was generated or developed at the City of Hope. It's been engineered a step further so that it forces tumor cells to produce what iNKT cells recognize, which is the CD19. And for the virus itself, or the virus that also produces a checkpoint inhibitor, it's actually started clinical trials already in 21 and 22, and Imugene continued to do great things. The FDA cleared their IND to start a combination study with onCARlytics and Blincyto, which is not a cell therapy, but a bispecific antibody. So it's good that the regulators are already familiar with the idea of a combination study with onCARlytics. And how this would look in practice is essentially the virus would be delivered to the patient with the cancer cells. That would actually then go and infect the solid tumors. So that's the orange and green virus infecting. Its then job is to replicate and multiply and burst and kill some of those tumor cells. For those it doesn't, it will label them on the surface with CD19 in green here. And then we're able to administer the iNKT cells, which will then come in and recognize that CD19 and destroy those cancer cells. Now, as I mentioned earlier at the beginning of the seminar, that iNKT cells also kill pro-tumor cells that sit and more difficult to treat the cancer, and also recruit other host components like T cells and NK cells. So we do see that there's a genuine multi-pronged approach having an impact in solid tumors. So I should just lastly point out, we progressed that data to the in vitro stage in January, February this year, and looking forward to getting into the animal models and presenting data in the second half of the year. So in terms of the next 6 to 12 months, as I said, we've had a pretty busy prior 12 months, but we are certainly looking forward to a strong finish to this year and an excellent start to 2024. So we do expect to advance ALA-101 into its first phase 1 human clinical trial. And we're also continuing to assess and review novel complementary technologies that we think expand the use of the iNKT cell platform to move into solid tumors. So in terms of milestones, we'll complete the process optimization and scale up in preparation to manufacture GMP grade product for clinical trials. We'll complete production of the GMP lentiviral vector. And while we're doing this, finalize the clinical trial plan for the phase 1 study. And first half of 24, we'll finalize the manufacture of the product for clinical trials and have the documentation being prepared and submitted to regulators like the FDA in the form of an IND or to the Australian TGA as an equivalent. And in terms of our platform itself, so we're excited to continue working with Imugene to combine the CAR-INKT cells with onCARlytics. As I said, we're expecting to get data in the second half of this year, and we're continuing to review additional CARs that we think could be beneficial for the platform, and as well as continuing to work on the technology that we have under option from the University of North Carolina. So for the first half of 2024, we'll continue to work through some of those proof of concept studies, and looking forward to getting some good data out for each of those. Now, in terms of our leadership team, as I mentioned, it continues to get stronger, and I won't go through every individual here, but just to say that it has, it's significantly strengthened even over the last 12 months. So it's a very high quality team with a lot of experience, not at one particular stage of drug development, all the way from early stage research into drug development and then through the commercialization of technologies. So looking forward to continuing to build that out where necessary. And I'd just like to summarize by saying, so we do have a very exciting CAR-INKT cell platform. We think this serves as an excellent platform as an off-the-shelf cell therapy treatment. Now, as I mentioned, we know that the CAR-INKT cells have multiple anti-cancer properties, both killing the cancer cells themselves, but also shaping the host in terms of the tumor microenvironment, as well as recruiting their immune systems. We're comfortable now that we've got a manufacturing process that enables us to produce multiple doses per batch. And we just think this goes a long way to improving logistics and patient access for these types of therapies. So we're excited that ALA-101 is starting to advance towards clinical trials, and we're expecting that to commence in 2024. We continue to build out our pipeline through strategic acquisitions as well as collaborations. And we do believe that for all these reasons that we've seen previously, but also now moving forward, that we still sit as an exciting company well positioned for growth. And I'd just like to finish with highlighting that we do have a share purchase plan currently open. As I mentioned a moment ago, we did have the placement closed a few weeks ago now. It was oversubscribed to raise 4.1 million, and the SPP offer price is same as the placement price at 4.5 cents, which I believe the current share price is trading slightly above that. And it does represent a 10% discount to the last closing share price prior to the announcement of the placement. Just reiterating that the closing date is the 29th of June, and shareholders are able to apply for up to $2,500 to $30,000 of shares for those shareholders that are eligible. There'll be no transactional brokerage costs for shareholders participating, and the funds raised under the SPP will enable us or be contributed towards progressing ALA-101 into phase 1 clinical trials with patients that have CD19 positive hematological cancers, as well as to strengthen our iNKT cell therapy platform and to cover the cost of the SPP as well as general working capital purposes. So with that, I'd just like to say, again, I think we've had a really strong start to the year. Certainly appreciate all the support we've received from our current shareholders, and certainly looking forward to a really strong 6 to 12 months ahead. And Matt, I'd turn it back over to you, and I'd be happy to take any questions.
Thanks, Michael. We'll move on to the questions as you've mentioned. I'll just remind everyone, if you have a question you want to send through still, please use the Q&A function within Zoom, and we'll get through as many as we can. Michael, the first one, you touched on it being a positive start to the year, and as I think many people know, it's been a tough sector in biotech. Why do you think Aravella's had such a positive start for the first half of the year?
Yeah, and you're right, Matt. It certainly has been a pretty tough market to watch. You're still seeing it quite a lot in the US. Many companies trading below cash value, so it is still a pretty tough sector out there. But look, I think from our perspective, I think we've worked very hard to stay focused on what it is that we think we're good at. It is working with iNKT cells. and bringing those through the manufacturing stages and into clinic. And I think the data that we presented at AACR, that did seem to culminate with a fairly large increase in the share price. So I think data at the end of the day, I think still speaks volumes. And again, having a nice outcome for the in vitro study with imaging was also a really big positive. And I think, again, building the team with people like Tom and Nicole coming on board, it's really looking like a bright future for a small Australian Stock Exchange listed company.
Thanks, Michael. One question that's come through, I think you might have touched on this before after the question came through, but if you want to recap anyway, can you please update on the in vitro and in vivo preclinical study on ALA-101 plus onCARlytic virus, progress plus milestones?
So we can't say too much about the data just yet, other than that the in vitro data was promising, and that's why we're excited to continue moving into the animal studies for the combination approach. And that's what we're doing exactly at the moment, working hard to produce the materials that we require, set up the studies in a manner that makes sure that we're very comfortable with the data. And then once we've got that, we'll certainly update the market when they're ready, but expecting that data in the second half of this year.
And then a follow-up to that was, when do you anticipate first patient dosing commencing in the ALA-101 phase 1?
Yeah, based off the trajectory that we've got to date. So we're working hard to get the manufacturing finalised through the second half of this year. And that's going well. We're making good progress. We'll get the componentry like the lentiviral vector finished before the end of the year. And then essentially going through the process of manufacturing the product, submitting what we need to the regulators. And so hopefully it would be close to around the first half of the year, starting second half of the year.
Thank you. Another question that's come through is, can you give some colour as to the effort and complexity that it takes to get into the clinical trials?
Yeah, absolutely. So look, it's quite a rigorous process, and I think for good reason. All regulators all over the world, including the TGA, they'll just want to make sure that whatever you're about to put into patients is safe. And that's fine by us. And of course, a really important part of it for cell therapy comes down to the manufacturing. And so that's why we've worked very hard to work with some of the best names globally for the lentivirus that we need. So that was a very good decision. And as I said, we'll get that before the end of the year. And then it's about plugging that into the process that we establish throughout the second half of the year. So that's going to be the biggest factor. And again, we recruited a gentleman only recently that came out of spent time at Kite Pharma, which is one of the probably the most successful cell therapy company, for exactly that reason. We want someone like that assisting us and helping us to manage that process carefully. But that's going to be the key piece of information. All of that ends up getting written up into what we call an IND, or an investigator brochure, which documents the preclinical proof of concept studies, the safety studies, as well as the manufacturing. And it's essentially just being able to produce that in a controlled fashion that will be really important. And once we've got that all in a row, we'll submit that to the regulators, either the FDA or the TGA, depending on which clinical pathway we'll take.
Thanks, Michael. And just one final one broadly, what can we expect from the company over the next 12 months?
So I think there's a number of good things to happen in terms of ALA-101. As I said, we've got a number of manufacturing milestones to tick off. And so looking forward to getting those done and announcing those as well. Looking forward to getting the imaging data with the combination in animal models in the second half of the year. And as I mentioned, we are looking to screen a number of different technologies that we think may be of interest for the company, and specifically for solid tumours. So looking to provide more updates on those as well as we move forward.
Thanks, Michael. I'll just hand it back to you to provide a bit of a closing comment.
Thanks, Matt. Yeah, just like to say thanks again to everybody for taking the time to dial in. As I said, we're very appreciative of all our shareholders and the support we've been shown, and we're looking forward to having an impact in the lives of cancer patients and also creating value for our shareholders. Thanks, Matt.